three models of DNA replication, okay? One is called, you get one called the conservative, and two called the semi-conservative, and three called dispersive. Okay? This is where another pen is required. So in conservative, you have the DNA strand like that, okay? Yeah, in conservative you have a DNA strand like that, which is our DNA, and that basically forms okay, and it also forms another one which is not made of any of the original strands, okay? So that's the original strands, and this is a new DNA that's not the original strands. In the semi-conservative, so you get I should have done that slightly differently, I think. I'm going to just redo that actually. Okay, so I'll redo that slightly. So you get, in the conservative, you got the helix made of two strands. You get one daughter forming made of the original, a new one made of new strands, okay? In the semi-conservative, you get semi-conservative, you get two different strands. You'll find that when I draw them different colours. So you get, in one strand you get an original strand, okay, forming with another strand, and you get a red strand forming with another strand, okay? And then you get dispersive, I can't spell dispersive, okay? So in this case you get two strands, okay, like that, and they result in two daughters which are basically like this. Okay, so you get okay, so that's three basic models for DNA replication, okay? So in this case, both original strands form a new double helix, and then you get a second double helix formed, but not using the original strands. In the semi-conservative you get two strands, one strand forms one strand of the new helix, then you get another strand formed, and the red strand forms a strand of the new helix and another new strand is formed. So that's the red one there and that's the black one there. So they, each of these strands goes to one of the daughters, and then a new strand is created. In dispersive you get two strands, but they kind of get mixed up, so you get red and black alternately. Okay, well the both strands get mixed alternately. Red, they actually get mixed. Okay. And then it just continues on. This that would form another one. So the same process occurs. Okay. All evidence, so I'm giving you three mechanisms here. All evidence shows the semi-conservative one. The semi-conservative model is how human and all lice DNA replicates, okay? Replay. Okay, this one is the correct one because each of these strands forms, the black strand gets converted, each of these strands goes to the new daughter and another strand gets made, and another strand gets made. This is wrong and this is wrong. These are just models for how it was thought it worked, sort of back in the 50s and 60s, 70s, but this one is the one that's correct. These are wrong, okay? But these are three different models of how DNA was thought to have replicated way back in the 1950s when its structure was first identified. Might even go back further. I can't remember the history of it, but that's the three models, okay? I don't remember enough details about the history to tell you more, but all evidence shows is this one that's correct. This is how human DNA replicates. It's how mouse and tree and daffodil and bumblebee and fish and swan, bird, bacterial DNA all replicate, okay? Semi-conservative. And even coronavirus. Well, actually coronavirus is an RNA, so it doesn't use this at all, but I guess it has similarities. There are some of the sort of the way coronavirus infects us and replicates has got connections to the way ours works, but it's not, because it's not actually a DNA, it's not quite this. It has to be made into DNA first. Now I won't go into it, but it's, I think it goes messenger RNA and DNA and then RNA, but anyway, yeah, all evidence shows is this one, okay? And these are wrong, okay?